Hello, I'm Dennis Grant, and today's lecture will cover some basic concepts in immunology and how we can pharmacologically manipulate the immune system with immunotherapeutic agents for the treatment of immune-related diseases. We'll briefly review the immune response and how pharmacology fits within this domain. In addition to eliminating pathogens, the immune system has been implicated in the pathogenesis of many diseases. And this has made it possible to treat these diseases by developing drugs that target the immune system itself. We'll discuss what constitutes the immune system and the different cell types and their functions, with an emphasis on the two major types of immune responses, namely the innate and adaptive responses, which are responsible for dealing with invading pathogens through differing mechanisms. Finally, we'll focus on some interventions that take advantage of the immune response to treat certain disease states, and we'll examine some specific immunotherapeutic agents and the mechanisms by which they elicit their effects. Immunology is a branch of biomedical sciences that studies the different types of host defense responses within our body. In general, the immune responses exist as a critical balance between immune activation and immune suppression. And if this balance is broken, the outcome may be harmful to us, such as is in the case of hypersensitivity and autoimmunity, when the immune cells attack normal body tissue. In fact, the immune system has been shown to play a role in many diseases. Immunopharmacology focuses on agents that modulate the immune response so as to favor a desired clinical outcome. This applies in cases when the immune system has already turned against itself, such as autoimmune disease, or where the immune response is downregulated in the immunosuppressive microenvironments of tumors in cancer, or where there is immune rejection in the graft versus host reaction, which is common in tissue transplantation. So let's start off with a brief anatomy of the immune system and how it protects us against pathogens and infections. The most basic defense is the skin and mucous membranes, which separate the internal environment from the external environment and prevent the entry of microorganisms into the body. This provides a very non-specific protection. Mucous membranes produce viscous mucus, which can trap foreign matter and pathogens. Invading pathogens that enter the respiratory tract are caught in the mucus and transported out of the lungs towards the mouth and either swallowed or spit out. Similarly, the acidic environment of the stomach produced by mucous membranes can kill many microorganisms. If a pathogen breaches these physical barriers, then the immune system tries to eliminate the invaders. Next is the lymphoid tissues, which are crucial for the production and maturation of various cell types that help initiate and propagate a variety of immune responses. The immune response is really divided into two main categories, which work together to protect the body against pathogens. Innate immunity, which is also referred to as nonspecific immunity, is the first line of defense against pathogens. In contrast, adaptive immunity, or specific immunity, represents a stronger defense against a specific pathogen, and it takes longer to develop since it requires immunological memory. Various immune cell types are produced and mature in lymphoid tissues. These lymphoid tissues can be subdivided into primary and secondary lymphoid organs. The primary lymphoid tissues are the sites of lymphocyte production and maturation, whereas the secondary lymphoid tissues are the sites where lymphocytes contact pathogens or foreign matter and become activated as part of an immune response. The main tissues of the primary lymphoid system are the thymus, the bone marrow, and the fetal liver, which are not shown in this particular figure. Bone marrow is the site where all leukocytes, including erythrocytes, develop from the same hematopoietic stem cell progenitors. B lymphocytes and most other leukocytes also mature in the bone marrow, whereas T lymphocytes migrate to the thymus to fully develop and mature. After the maturation stage, B and T cells migrate from primary lymphoid tissues into secondary lymphoid tissues, where they can come into contact with foreign substances, become activated, and initiate an immune response. Secondary lymphoid tissues include the spleen, lymph nodes, tonsils, adenoids, appendix, lymph nodules of the GI tract and the lining of the GI tract in areas called Peyer's patches, and they're dominated by B cells, T cells, and macrophages. Cells of the immune system begin to differentiate into different cell types in the bone marrow. This is where pluripotent hematopoietic stem cells can differentiate into either myeloid or lymphoid cell progenitors. Myeloid cells are comprised of granulocytes, monocytes, and macrophages. Granulocytes consist of neutrophils, eosinophils, and basophils, and these have cytoplasmic granules. In contrast, lymphocytes and monocytes lack granules. In addition to these cells, macrophages, mast cells, and dendritic cells are also derived from the myeloid stem cells. Phagocytes are also myeloid cells, and they comprise the class of cells that engulf foreign particles, and by so doing, remove microorganisms from the blood or tissues. Neutrophils, eosinophils, monocytes, macrophages, and dendritic cells are all phagocytes. Neutrophils are the most abundant and are particularly involved in fighting bacterial infections. Eosinophils tend to defend against parasites by releasing toxic substances. Monocytes phagocytose in the blood, and macrophages are 5 to 10 times larger than monocytes. They have greater phagocytic activity, and they're found in almost every tissue. Looking at the lymphoid progenitors, lymphocytes provide an immune system with diversity, specificity, memory, and the ability to distinguish between self and non-self. The three main types of lymphocytes are B cells, T cells, and natural killer or NK cells. B lymphocytes have the ability to develop into plasma cells upon contacting foreign molecules, which are also known as antigens, and as plasma cells they secrete antibodies that target the antigen for destruction. In contrast, T cells do not directly damage foreign molecules, but upon contact with an infected cell, over several days a T cell develops into a cytotoxic T cell, which can then destroy the infected cell through the release of mediators such as perforin and granzyme B. NK cells are important in fighting viral infections. NK cells kill by the same mechanism as cytotoxic T cells, but they tend to respond before B and T cells, making NK cells an important part of the early innate immune response as well. So let's review the inflammatory response and its implications for whether the immune system will be triggered to develop a more specific response or not. In the event that a pathogen infiltrates through skin or mucosal barriers, the first response would be the onset of inflammation. There are 5 steps of inflammation. In Panel A, after tissue injury like a cut or bacterial infection, macrophages engulf debris and matter. Panel B illustrates that capillaries dilate and become permeable to allow a greater flow of blood into the affected area. And this way, cells of the innate immune system can get easy access to pathogens. In Panel C, the presence of macrophages in the affected area contains the foreign matter from spreading further into the body. Panel D demonstrates the process of recruiting cells to the affected area as leukocytes continue to migrate into the area. Finally, in Panel E, there's recruitment of leukocytes, mainly neutrophils, which continue to clear the infection, and an eventual resolution of the inflammation will occur. These events are nonspecific, meaning that inflammation proceeds in the same way, regardless of the trigger involved. However, they do involve immune cells primarily from the innate immune system, such as macrophages and neutrophils. The innate immune system provides a rapid defense and recognizes most pathogens, but it's still a nonspecific response. The most common cell types are the granulocytes, which consist mainly of neutrophils, basophils, and eosinophils. Macrophages and monocytes are a major cell type of the innate immune system, as are NK cells, which act at the interface between the innate and the adaptive immune responses. Pathogens are recognized through common structures on their surface, which are referred to as pathogen-associated molecular patterns, or PAMPs. Many cells of the innate immune system have specialized receptors that bind PAMPs and initiate a defensive response. Aside from the physical barriers of the body, the inflammatory process and interferon proteins, natural killer cells are also a very important part of the innate immune system. As their name implies, these cells can kill other cells. NK cells recognize infected cells and release perforin, which opens pores in cells and kills them by allowing the cells to take up more ions and water, which makes the cells swell and eventually burst. NK cells are activated by interferons and cytokines released by activated macrophages, such as interleukin-12. NK cells protect against viral infections, tumors, bacteria, and protozoans. They can also play an important role in the adaptive immune response because they can be activated by T cells, and we'll talk a little more about this in the coming sections. The complement system acts as part of the innate and adaptive immune responses, depending on the presence or absence of antibodies. The complement system consists of 30 plasma proteins that destroy invading microorganisms, such as bacteria, and it can be activated in 3 ways. The first is through binding to antibodies that are attached to the bacteria. This is referred to as the classical pathway. The second is through complement lectin molecules binding to mannose-containing carbohydrates on bacteria. And the final path, which is called the alternative pathway, is through the direct binding of a complement protein to carbohydrates that are present on the surface of bacterial cells. All of these pathways converge and generate the same set of complement proteins, which protect against infections through 4 different mechanisms. By covalently binding to pathogens and marking them for destruction by a phagocyte, by recruiting phagocytes to the infection site, by histamine release, and by creation of a membrane attack complex, or MAC, which forms pores that pierce the bacterial cell wall. The adaptive immune system is more efficient and highly selective, and it comes into play after the initiation of innate responses in which the pathogen or antigenic insult still persists. Some key characteristics of the adaptive immune system are outlined on this slide. It's specific because the response is mediated by lymphocytes, which means that specific cell types attack specific pathogens and eliminate them. It's diverse because these cell types can recognize a multitude of varying pathogens. Moreover, it has immune memory, which remembers antigens that it has already encountered and can distinguish non-self antigens from self-antigens in a way that prevents autoimmune reactions when it's functioning properly. The 2 major cell types involved in adaptive immunity are the B and T lymphocytes. Pathogens or antigens that survive the innate immune response will come into contact with B and T lymphocytes. The adaptive immune response, mediated by these cell types, is divided into 2 main parts. The humoral response is mediated by B lymphocytes, and it produces antibodies. It mainly responds to bacteria, toxins, and viruses. The cell-mediated response is mediated by T lymphocytes, and it affects bacteria, viruses that are hidden in and inside infected cells, transplanted tissues, and cancer cells, which are perceived as being foreign. Let's take a closer look at the first characteristic of the adaptive immune system, which is specificity. As illustrated on the right-hand side of this slide, both T and B cells bind foreign substances that are known as antigens. Each antigen has a unique structure, and it contains a different recognition site which can be detected by lymphocytes. B and T cells recognize each antigen through antigen receptors that are situated on their surfaces. B lymphocytes produce antibodies against the pathogen. Antibodies are Y-shaped molecules, and they consist of 4 chains, as illustrated here on the top left-hand side of this slide. The variable regions are called this because the sequence of amino acids varies significantly from antibody to antibody. The 2 regions of the light chain form 2 identical antigen binding sites, and they can bind to 2 sites on a given antigen. Each antibody binds specific antigens, just like an enzyme substrate interaction. There are 5 types of heavy chain which serve as the basis of 5 different classes of antibodies, and these will be discussed in the section on humoral immunity. The antigen receptor on the T cell is called the T cell receptor. These are different in structure from antibodies, and they act only as surface receptors for antigen recognition. Each B or T lymphocyte has approximately 100,000 antigen receptors, each with the same antigen specificity. The receptors are determined by random genetic events that generate a phenomenal array of B and T lymphocytes, each with the ability to bind to one specific antigen. This allows for the recognition of millions of possible antigens, even though they've not been exposed previously to the antigen. Each microorganism only activates lymphocytes that have the specific receptor for that antigen. This antigen-driven activation of lymphocytes is called clonal selection. Differentiation of lymphocytes in the presence of antigen can give rise to 2 identical cells that are called clones. The first group of cells are the effector or plasma cells that are short-lived and combat the antigen right away. The second are memory cells, which are long-lived and have membrane receptors for the same antigen. These memory cells allow for faster elimination of the antigen in case it reappears, since the memory cells, upon reactivation, can quickly give rise to thousands of cells with specificity for the same antigen. Immune memory is acquired following exposure of the adaptive immune system to the antigen and subsequent response. The primary immune response occurs when an individual is first exposed to antigen, and the antigen induces changes in lymphocytes to induce a response. The graph on the left shows this primary response, where antigen-selected B and T cells proliferate and differentiate into effector cells. This process takes time, and initially one might feel ill, because it takes a while for the antibodies and cytotoxic T cells to develop and combat the pathogen. However, upon re-exposure to the same antigen, generation of an acquired immunity and secondary immune response will occur to combat the same antigen at a much faster rate and a far greater magnitude of effect. This secondary response, demonstrated on the right, occurs as a result of immune memory, which is acquired during first exposure to the antigen through production of long-lived clones of memory T and B cells, and these can proliferate much quicker. In addition, self-tolerance is the process by which B and T cells do not react against self. During B and T cell maturation in the bone marrow and the thymus, lymphocytes with antigen receptors that have the potential to recognize self are eliminated through programmed cell death, or apoptosis. This process distinguishes self from non-self, and it prevents the induction of an autoimmune response. Humoral immunity involves the role of B cells and the elimination of pathogens. Once a B cell encounters an antigen, it responds in two ways. The first is by producing short-lived plasma cells which are secreted rapidly over a period of 4 to 7 days. The second is by forming memory B cells. Plasma cells secrete more antibodies which target pathogens directly for destruction, while memory B cells, as described already, are more long-lived and lead to a faster immune response if the pathogen reappears again in the future. Antigens that provoke the production of memory B cells and plasma cells are known as T-dependent antigens because they require a special cell type called a T helper cell. The T helper cell, upon encountering an antigen, releases cytokines such as interleukin-2 or IL-2. Without IL-2 production, most cells would develop into plasma cells and not memory B cells. So without IL-2, there's no immunological memory. In contrast, some antigens, such as polysaccharide antigens, can activate B cells without T cell help and are known as T-independent antigens. After antibodies are produced, there are 5 major classes of antibodies that vary in structure and in function. Antibodies are involved in 2 main functions in the body. They bind specifically to an antigen, triggering the disposal of the antigen. The tail shape of the Y is responsible for disposal, while the antigen binding site is responsible for recognition of the antigen. 5 different classes of antibodies exist based on the structure of the tail, and these are described in this figure. These antibodies differ in structure and in functions, and also in their distribution. For example, IgM is a complex of 5 Y-shaped antibodies. All classes of antibodies can participate in processes such as neutralization and agglutination, while some more specialized classes also participate in opsonization, complement activation, and stimulation of NK cells, as summarized in this table. The major antibody type found in the serum is IgG. IgG and IgM are the major types of antibodies that are involved in complement activation. IgE is mainly found during the occurrence of allergic reactions. IgA is found mostly in epithelial cells, while the function of IgD is mostly unknown, and it has very low concentrations in serum. We mentioned in the previous slide that the main purpose of antibodies is to bind and dispose of the antigen. This happens through the 5 steps that are outlined on this slide. Neutralization occurs when an antibody blocks antigen activity by binding to it and rendering the antigen inactive. An example is bacterial toxins, whereupon antibody binding to the bacteria stops production of the toxins. Agglutination happens when antigens are clumped together by thousands of antibody molecules and are then neutralized. This is possible since each antibody has at least 2 antigen binding sites, which can bind at least two antigens linking them together. When this process happens, antibodies form a large complex with the trapped pathogens, and this neutralizes the pathogens and makes them more susceptible to elimination from the body. Opsonization occurs when an antibody-bound pathogen is phagocytosed. The binding of antibody to a pathogen renders it more susceptible to phagocytosis because antibodies can bind both antigens and phagocytic cells on specific receptors and trigger the phagocyte to engulf both antibody and the antigen, thereby eliminating the threat. Complement can be activated by IgG and IgM molecules. Once these antibodies bind to antigen, they can initiate the complement activation cascade, which leads to the creation of pores on the pathogen and ultimately destruction. Activation of NK cells occurs when IgG antibodies enhance the nonspecific killing action of NK cells. Antibodies mark the cell for death, and NK cells have receptors that bind the antibody, making the killing activity of NK cells more specific. When this happens, the NK cells can release perforin, which opens holes in the cell membrane and kills the infected cells. Antibodies defend against foreign substances in the blood and lymph and cannot access antigens that are located inside cells. T cells, however, can make contact with and respond to cells that are infected with a foreign substance or are abnormal. There are three major classes of T lymphocytes: helper T cells, cytotoxic T cells, and suppressor T cells. Helper T cells secrete cytokines that enhance the activity of B cells, cytotoxic T cells, suppressor T cells, and themselves, and they also enhance the actions of macrophages and NK cells. Cytotoxic T cells are directly responsible for cell-mediated immunity, and they kill infected cells. Suppressor T cells produce cytokines that suppress the activity of B cells, helper T cells, and cytotoxic T cells. These cells are not well understood, and recently new types of these kinds of cells have been identified. Classical suppressor T cells are ones that express the transcription factor FOXP3. All of these cell types have the T cell receptor, which identifies foreign antigens. However, in order to detect the antigen needs to be associated with a special class of self-identifying proteins known as the major histocompatibility complex, or MHC molecules, which are located on antigen-presenting cells. To elicit a T cell response, antigen must be bound to an MHC molecule on the surface of a cell as a complex. This process, known as antigen presentation, is recognized by T cells. The MHC is encoded by 100 genes on chromosome 6. In addition, each person has a unique MHC called HLA, standing for human leukocyte antigen. This is very important because it explains why some people are susceptible to certain reactions or allergies, while others are not. The MHC is responsible for a variety of immune responses, including rejection of transplanted skin grafts and organs foreign to the recipient, because the MHC molecule has antigens that are attacked by the recipient's immune system. Two classes of MHC exist, and they differ in which cells express them to allow the immune system to distinguish self from foreign matter. MHC1 is found on the surface of almost every cell, but it's found in greater levels on the membrane of leukocytes. They present endogenous antigens to cytotoxic T cells, which are also called CD8 positive cells due to the expression of the MHC co-receptor glycoprotein CD8 on their surface. MHC2 is found only on a few specialized cell types like macrophages, dendritic cells, activated B cells, and cells in the thymus. All of these cells are referred to as antigen-presenting cells because they usually engulf the antigen, break it apart, and present it to the surface of the cell with the help of MHC2. MHC2 presents exogenous antigens to helper T cells, also called CD4 positive cells, due to the expression of the glycoprotein CD4 on their surface, and these are then taken up into cells by phagocytosis. The process for antigen presentation to T cells is illustrated in this figure. First, the pathogen is broken down into peptides. As the MHC makes its way to the surface of the cell, it captures the antigen and carries it out to the surface of the cell. Class I MHCs capture antigens in infected cells and present them to cytotoxic T cells. Class II MHC bind to internalized antigens and transport them to the cell surface for presentation to helper T cells. Once the antigen is presented to the surface and recognized by the T cell receptor of either CD4 or CD8 positive T cells, then the T cells become activated. In general, this involves the release of various cytokines that recruit more immune cells to the site of infection and help clear the threat, or it leads to a direct response where the activated cell kills the pathogen immediately. Helper T cells are central to the coordination of immune responses. Activation of helper T cells involves two events. Firstly, helper T cells first bind with class II MHC foreign antigen complexes on the surface of macrophages, dendritic cells, and/or B cells. The helper T cells then receive from these cells an inducing signal in the form of IL-1. This makes the helper T cells proliferate and differentiate. This process can take up to 17 days to develop, especially on first exposure to an antigen. Activation of helper T cells can result in the release of several cytokines that activate other T cells, B cells, macrophages, mast cells, NK cells, and hematopoietic stem cells. Cytotoxic T cells have the ability to kill when they become activated. Two events have to occur in order for this to happen. Number one, the cytotoxic cell has to bind with MHC1 antigen complex on the surface of infected cells. And secondly, it also has to receive an inducing signal in the form of IL-2 from the helper T cell. Once activated, cytotoxic cells release perforins, which lyse cells. Overall, effective immune responses arise from multiple direct and indirect interactions among macrophages, helper T cells, B cells, and cytotoxic T cells. Now let's move on to a discussion of drugs that are used to modulate the immune system in order to obtain a therapeutic effect. The drugs that we'll discuss are ipilimumab, basiliximab, alefacept, etanercept, and several IL-1 antagonists. It's interesting to note that the first two drugs on this list, ipilimumab and basiliximab, are both monoclonal antibodies. And this is reflected in the mab ending to the names, meaning M for monoclonal and AB for antibody. In the clinic, several agents are used to suppress the immune system. These are used in autoimmune diseases and in organ transplantation. Several of the drugs that we'll discuss fall into this category. For an immune response to occur, two signals are necessary, which are shown in the left panel as 1 and 2. We already discussed signal 1, which is binding between the antigen on the surface of MHC to the T cell receptor. In the absence of a second signal, the immune response is called anergy, also referred to as tolerance, which puts the T cell in a quiet and non-responsive state. The second signal involves ligation of co-stimulatory molecules, CD40, CD80, and CD86, on the antigen-presenting cell to their respective ligands. As an example, CD40L is the ligand for CD40. As shown on the right panel, drugs like ipilimumab, batacept, and alefacept interfere with the second signal step. Activation of T cells is regulated through a negative feedback loop involving a molecule known as the T lymphocyte-associated antigen 4, or CTLA4. Following engagement of CD28 with CD80 or 86, CTLA4 in the cytoplasm makes its way to the cell surface, where it outcompetes or displaces CD28 for binding to CD80-86. This results in the suppression of T cell activation and proliferation. This property of CTLA4 has been exploited in cancer treatment, where an antibody that binds CTLA4, ipilimumab, prevents its association with CD8086. By so doing, the antibody drug ipilimumab prevents the T cell from entering an anergic state, and the active state of T cells is sustained. Ipilimumab is a monoclonal antibody that targets the human CTLA-4 surface marker. It has been approved since 2011 for the treatment of melanoma, which is an aggressive form of skin cancer. In clinical trials, ipilimumab resulted in improved patient survival. Notably, this antibody has a long half-life of around 2 weeks, which reduces the frequency of drug administration to about once every 3 weeks. The immune response is important in cancer. Here we focus on the cytotoxic T cell, abbreviated as CTL in this particular picture. These T cells can initiate immune responses and cause tumor lysis. In some types of cancer, tumors are able to evade immune responses. Immune modulating drugs such as ipilimumab can boost anti-tumor immunity. Cancer cells have learned how to evade immune responses that would eliminate them. For example, by stimulating the upregulation of CTLA-4 on T cells. Since CTLA-4 binds with higher affinity to B7, it competes for binding and results in the downregulation of IL-2. Lymphocyte recruitment into the tumor is diminished. As shown in panel C, ipilimumab binding to CTLA-4 blocks the binding between CTLA-4 and co-stimulatory molecules. The action of ipilimumab allows for the activation signal between CD28 and B7 to be reestablished, IL-2 to be produced, and lymphocyte recruitment into the tumor. Interfering with immune checkpoints in cancer therapy can have undesired consequences on other normal tissue cells because blockade of CTLA-4 can compromise immune tolerance in normal tissues. The body's immune response may become hyperactive towards normal tissues and result in autoimmunity. A unique set of toxicities referred to as immune-related adverse events has been seen with anti-CTLA-4 antibodies. These events are dose-dependent. By reducing the dose of antibody, one can often reduce the adverse events, but this can also reduce tumor kill. Basiliximab is a chimeric mouse-human IgG antibody that's used to prevent transplant rejection. This antibody has a long half-life of 1 week, and levels that saturate its target, the IL-2 receptor, are readily maintained. Basiliximab, combined with other immunosuppressant drugs such as cyclosporin or corticosteroids, can have a synergistic effect in inhibiting IL-2 signaling, since cyclosporin inhibits the transcription of IL-2. Basiliximab is effective for pancreatic transplantation, and it's generally a very safe drug. Basiliximab acts by preventing binding of IL-2 to its receptor. Remember that activation of T lymphocytes is mediated by binding of IL-2 to its receptor, IL-2R. IL-2R is a heterotrimer in structure, and it's composed of 3 subunits: the IL-2R alpha, IL-2R beta, and IL-2R gamma. In order for the T cell to be activated, IL-2 must make contact with all 3 subunits of the IL-2 receptor, as shown in the diagram. Basiliximab binds to the IL-2 alpha subunit, thereby preventing IL-2 from binding to the whole complex. This results in inhibition of signal transduction cascades that would normally lead to immune responses. Alefacept is a genetically engineered immunosuppressive protein that's used in the treatment of psoriasis, an immune-mediated skin reaction in which the immune system recognizes a normal skin cell as an immunoreactive cell type. The inflammatory environment during psoriasis is associated with an increase in CD4 and CD8 positive T cells. Alefacept binds to the CD2 surface marker on T cells and interferes with the binding between T cells and antigen-presenting cells. In this way, it inhibits activation of T cells by inhibiting signal 2, the critical second step of the immune response that we discussed earlier. Alefacept targets T cells and NK cells, and it induces apoptosis of CD2 positive cells, thus reducing T cell numbers. This drug does not inhibit primary or secondary antibody responses to foreign antigens, suggesting that patients taking Alefacept are not at higher risk for infections. Etanercept is a dimeric fusion protein made up of two soluble human tumor necrosis factor, or TNF, receptors linked to the constant Fc region of human IgG1. This drug acts as a cytokine inhibitor by reducing serum levels of TNF-alpha, which is a pro-inflammatory cytokine. Other TNF inhibitors have also been developed, and they all bind to TNF. TNF inhibitors are useful to treat inflammatory diseases such as arthritis. Etanercept has a short half-life because it's a fusion protein. It's used for arthritis, but its immunosuppressive properties can cause autoimmune diseases and worsen pre-existing infections. As mentioned above, TNF is a pro-inflammatory cytokine produced by a variety of cells such as lymphocytes, macrophages, and NK cells. TNF can bind to two receptors, TNF receptor 1 and TNF receptor 2. Binding of TNF to its receptors causes an extensive inflammatory response that includes the production of IL-6 and IL-1, upregulation of major histocompatibility complexes, chemokine release, and upregulation of adhesion molecules that are involved in leukocyte activation and migration. All of these processes lead to complement activation and antibody-mediated or cell-mediated cytotoxicity, which can lead to inflammation and tissue damage. Etanercept binds to both TNF-alpha and TNF-beta and inhibits TNF-mediated inflammation. Etanercept can prevent TNF from binding to its receptor. Upon stimulation of TNF-producing cells, there's an increase in the surface expression of transmembrane TNF, denoted here as TM-TNF. TM-TNF can be cleaved by the TACE or TACE enzyme, and it's released outside the cell as soluble TNF or sTNF. sTNF or TM-TNF can bind TNF receptor 1 or 2. Binding causes activation of NF-kappa B and transcription of inflammatory genes. Etanercept can block the action of TNF by binding either to the soluble form or the transmembrane form of TNF. When TNF does not bind to its receptor, NF-kappa B signaling is blocked and inflammation is reduced or prevented. IL-1 is a pro-inflammatory cytokine produced by macrophages, monocytes, B cells, and NK cells. The drugs anakinra and canakinumab prevent inflammation by preventing IL-1 from binding to its receptor. Anakinra is a recombinant form of the naturally occurring IL-1 receptor antagonist that prevents IL-1 from binding to its receptor. It's used to reduce pain and swelling that are associated with rheumatoid arthritis. Canakinumab is a recombinant human anti-IL-1 beta monoclonal antibody. It binds human IL-1 beta and prevents it from binding to its receptor. IL-1 beta can be produced through an innate immune response. IL-1 beta is produced as an inactive 31 kilodalton precursor called pro-IL-1 beta. This is mediated through toll-like receptor signaling, which recognizes pathogen-associated molecular patterns, or PAMPs, and responds by inducing transcription of the pro-IL-1 beta gene. After the production of pro-IL-1 beta, a second step converts the inactive pro-IL-1 beta to the active 18 kilodalton form. The conversion is mediated by a complex called the inflammasome. Activation of NLRP3 leads to proteolytic cleavage and activation of caspase-1, which then cleaves pro-IL-1 beta to IL-1 beta, which is then secreted from the cell. IL-1 activates a number of cell types such as B and T cells and NK cells, as shown on the figure to the right. This leads to an inflammatory environment and induction of arthritis. These IL-1 antagonists block the inflammatory pathway. So in summary, in this lecture we've discussed how the immune system is structured and how it functions to protect us from pathogens. We've discussed nonspecific physical barriers as well as innate and adaptive immunity. Complex immune responses can be manipulated with drugs in order to fight various diseases such as cancers and inflammation. However, manipulation of the immune response frequently results in side effects that are related to immune activity. Our current understanding of how immune responses are initiated and resolved remains incomplete. However, basic research into immune system function and immunopharmacology drug development represent rapidly expanding and clinically significant areas. Thank you for joining me in this discussion of the immune system and the use of drugs to pharmacologically manipulate the immune response.